Welcome everyone. Hello. My name is Simon. I am the coordinator of the project ProxIMed. Welcome everyone to this webinar. I will shortly share my screen before we start this event. Alrighty. Yeah. Okay. So welcome again everyone to the ProxIMed webinar about insights from ProxIMed's young researchers. Um, I'm very happy that we have many participants that are interested in how it is to do research in a European project. Before we start, I have to give you a short disclaimer. So this meeting will be recorded, and as it is part of a European funded project, the recordings might be published on our website, social media, and if you don't feel comfortable opening up your camera, you are free to close your camera, use the chat. My colleague Nietzsche Erkan will keep track of the chat if there are any questions and you don't feel comfortable raising them yourself. We can raise them. And this is just for you to know that this meeting is recorded. If you don't consent to this meeting being recorded, I kindly have to ask you to leave because we are more or less required to record and disseminate this this webinar. All right. So what will be happening today? I will open up the dialogue and will give you a brief introduction about the project ProxIMed, and then we have uh, 8 great presentations from our young researchers within this project. I am more than happy that we have many female presenters, as it is also a part of the project to have some kind of woman empowerment and to have gender equity. Therefore, I'm very happy that we have many female presenters today, as well as having them in the project. So here you can see the topics. I will introduce the speakers during the presentations. And at the end, there will be a question and answer session. This means if you have any questions, I kindly ask you to write them down or write them in the chat. If you have specific questions for speakers, and we will do all of the questions at the end and the question and answer round. At the very end of the meeting, I kindly ask you to evaluate this webinar. I will share a link as well as I will share a link that you consent to the recording. All right. So what is ProxIMed in general? So we are exploring and implementing products with alternative proteins in the Mediterranean region. This means transforming the protein landscape in the Mediterranean area. And what is our mission? Our mission is to revolutionize the protein consumption in the Mediterranean region through innovation and sustainability. So we are trying to get products into Mediterranean diet. And therefore we focus on developing 20 alternative protein products with the active involvement of consumers, industry partners, and innovative technologies, which I will give an overview shortly, and our presenters will cover most of this. And of course, environmental impact, as well as improving food security and enhancing food choices, is a big topic within ProxIMed. And we strive to give people the opportunity for a healthier and more sustainable diet and future for the Mediterranean population. All right, so who are we? So we are 17 partners from 10 countries. Which are located around the Mediterranean area. We are some kind of a unicorn, as as we are in Germany. And here you can see our 17 partners. So we have partners from Germany, Turkey, Greece, Lebanon, Tunisia, Egypt, Spain, Portugal, also Malta, and not to forget Italy. Um, Please meet also the inventors, because without Professor Özlem Özmutlu and Professor Mecit Öztop, this project wouldn't have taken place, as well as the Prima Project Foundation, which is funded by Horizon 2020. Both of them, Professor Özmutlu and Professor Öztop, secured the project and were able to convince the funding service that the project gets approval. So what we do exactly? So we extract proteins from novel sources, agricultural byproducts, but also traditional plants that are already present in the Mediterranean diet. We use novel but also conventional processes to decrease the environmental impact, create effective processes, and improve functional properties while ensuring food safety and food quality. And to prevent to act in a theoretical scenario, ProxIMed also implements market research to act according to consumer needs. And those needs are then implemented into the product development of 20 products, which are then launched on Tunisian and Turkish markets. And therefore, we work closely with small and medium businesses and encourage them to serve the markets, besides encouraging startups to use those techniques and products we develop. And here you have a short overview, which are the raw materials that we use. So we have byproducts such as tomato pomace, tomato leaves, but as previously mentioned, traditional food materials such as lentils or sesame cake, and alternative proteins such as mycoprotein and insects. So this now brings me back to our agenda, and I am happy to introduce to you our first speaker, which is Ayşe Sultan Akgün from the Middle East Technical University in Turkey. So, Sultan, the stage is yours.
Thank you so much. I'm sharing my screen. Can you see it now?
Yes, perfect.
Okay, perfect then. Hello everyone, welcome to our webinar. I hope you enjoy and learn from this special session. I am Sultan, PhD student at Middle East Technical University in Biotechnology Department. I have food engineering background and I had my bachelor's and master's degree from food engineering department. I can position myself as a researcher and an entrepreneur. During my PhD, I have worked in many projects like Such Equality, Phantom, ProxIMed's, and two-step mushroom project. So I can tell that I'm trying to learn many topics as possible as I can. So metaphorically, I'm juggling between each project since I'm a multitasking person. One of the good sides of contributing many projects is traveling. This is me, and traveling keeps me motivated. This is our beautiful campus in Ankara, which is capital city of Turkey. Our university is one of the most prestigious university in Turkey and a technical university, which means we have lots of engineering departments, and one of them is food engineering. As you can see in this picture, this is our departments. What we do as food engineers, we involve every part of food production from farm to fork. The products that you see in the market shelves have been created by many food engineers. And this is our lab. and lab members. We are a big family in here. Each of us specialized in different topics like fermentation, alternative proteins, functional foods, emulsions, aerogels, chromatography, NMR, NIR, hyperspectral imaging, and many more. If you have a chance to visit our lab, you're very welcome here. How I started to my ProxIMed journey with a huge excitement because it was a new topic for me, which creates new opportunities. I was already working in fermentation field, but now I involved alternative proteins world. I'm working on producing single-cell protein from tomato pomace, which is a byproduct of tomato processing industry. But why tomato pomace? Because globally, 5.4 to 9 million tons tomato pomace is produced annually, depending on the volume of tomato production each year. And Turkey is the fourth biggest tomato producer in the world, which means huge amount of tomato pomace is generated. I use tomato pomace comes from tomato processing like tomato sauce production, tomato paste, which is very common to use it in every dishes in Turkey. What can be done with this tomato pomace? There are many studies in the literature you can find. Mainly, tomato pomace is used for tomato seed oil, lycopene, and dietary fibers and pectin production. It can be used also for tomato seed protein production, but I'm using it as fermentable sugar source for single-cell protein production. I aim to increase the protein content of tomato pomace. My fermentation journey was started with my master's degree. I was working on cellulase enzyme production from grape pomace, which is a waste of wine processing. So besides fermentation, I'm also passionate to transform industrial food waste into value-added products like protein, enzymes, and bioethanol for sustainable reasons. And our ProxIMed project is addressing many sustainable development goals. How can we produce single-cell protein? Let's cover it step by step. First, we need to obtain fermentable sugar. For this purpose, we need to apply pretreatment and hydrolysis methods. I wanted to use sustainable ones, so I started with hydrothermal pretreatment and continue with enzymatic hydrolysis. Then we need to filter and prepare our fermentation media. After obtaining our fermentation media, we need to sterilize it. Third step is growing the microorganisms. In my research, I'm using Saccharomyces cerevisiae as yeast and grow it in yeast extract peptone dextrose solution. Then here is the fun part. Fermentation starts. In bioreactor, we can control the temperature, pH, dissolved oxygen, agitation. And you can arrange your parameters according to your microorganism's growth condition. So we know that for biomass growth, we need high oxygen level. And during fermentation, I monitor it carefully. My fermentation parameters are in 3-liter bioreactor using 10% tomato pomace as carbon source at 30 degree, pH 5, 200 RPM, and fermentation time is 7 days. And finally, collecting the biomass by centrifugation. After collecting biomass, for further analysis, you can freeze dry and store them. For the analysis, every 20 hours, I took samples and checked optical density for the growth, reducing sugar content and soluble protein content. When I obtain my biomass and check its toxicity, amino acid content, allergenicity, and nucleic acid content, we can develop high-protein vegan products. First step, I'm sorry. The initial results of my fermentation was like this. In 24 hours, we see significant increase in microbial growth and decrease in reducing sugar content. So we can say that even in 20 hours, we can increase the protein content in huge amounts. The first step of my research was about the valorization of tomato pomace to use it as a substrate in fermentation. And I tried different hydrolysis methods, and output of this research was presented in World Processing Tomato Congress. The full text is accepted to publish, and it's accepted by ISHS articles. And many more publications will come about the protein production and fermentation parts. This is our ProxIMed's method team. While doing our research, we don't forget to have fun. And this picture is from our last meeting in Porto. We are happy to be a part of ProxIMed team. Thank you and see you next time.
Thank you very much, Sultan, for this comprehensive overview. I will open up the question then during the question and answer round. If there is anyone there who wants to ask a question, you can also write it in the chat for now so that we don't miss it. For now, I would like to open the floor for our next presenter, which is Bodoor Aljurf, also from Middle East Technical University in Turkey. Bodoor, the stage is yours.
Can you see my screen?
Yes, we can see it.
Okay. Okay. Hello, everyone. My name is Bodoor Aljurf, and I'm a master's student at Middle East Technical University. Today, I will be presenting my research topic, which is the innovation protein extraction from tomato pomace using ultrasound and microwave-assisted extraction and seeing their effect on protein yield and functionality. So let's continue. But first, before we get into technical details, I would like to introduce myself briefly. I have been part of the food engineering department at METU, Turkey, since 2018 when I joined as an international student. My passion for this field comes from my love for chemistry and mathematics, and I wanted to join an engineering program, and food engineering seemed to fit best. And also, it made me really interested to see how science can help to explore, can help to improve food quality, safety, and sustainability. Over the years, I have been involved in several projects that shaped my current work and made me reach to this point. The first two projects were in my undergraduate year, where I was an undergraduate researcher. First project is the crystallization of sucrose using microwave vacuum technique, and the second one is the production and characterization of intragastrically gelating Pickering emulsions. And then the third project was when I started my master's in 2022, and it was about increasing the storage time and food safety level of ground beef using 405 nanometer LED light technology implemented in refrigerators. All of these experiences led me to my current research project, which is the ProxIMed project. I don't want to go into details about what the project is about, but I will just give a brief introduction. The ProxIMed project focuses on exploring and implementing alternative protein sources in the Mediterranean region, and my research specifically focuses on extracting proteins from tomato pomace and tomato leaves and using innovative techniques to improve efficiency and functionality. And in this presentation, I will be focusing on tomato pomace. These are a few pictures from the experiments over the past months or even more than a year, because in the end, you will see all of the results in a table or graph, but it really took so much time and so much effort to be able to get to those numbers. And these are some pictures with the Ostoplab team. And the one in the middle is from the ProxIMed meeting in Izmir. And they were taken over 3 years. So let's start. First, I would like to discuss why are we always talking about alternative proteins? Why alternative proteins? There are a few key reasons. Global population growth. We need sustainable food sources to meet increasing demand. Climate change impact. Reducing greenhouse gas emissions, land usage, and water consumption is crucial. Food security and ethical concerns. So in summary, alternative proteins play a key role in building a more sustainable and resilient food system. And then why tomato pomace? My colleague mentioned it. just a few minutes ago, but I will be repeating the same points. Again, Turkey is one of the largest tomato producers, generating large amount of byproducts, and tomato pomace is rich in protein, fiber, and antioxidants. So utilizing these byproducts supports sustainability and reduces food waste. And potential applications include protein-rich food ingredients and functional additives. So by extracting proteins from tomato pomace, we add value to food industry waste and create sustainable food solutions. I don't want to go into too much detail about the ways we are extracting proteins or the methods of extraction, but there are a lot, and there are dry extraction methods, wet extraction methods. However, my main focus is on alkaline extraction, isoelectric point precipitation, which I will be referring to as the conventional method, as well as microwave and ultrasound-assisted extraction. Again, I don't want to go into detail about the mechanism. However, I will just be giving a quick introduction. In all these methods, the main goal is to extract protein. However, they work differently, and each of these methods have different efficiencies and impacts on protein yield and functionality. In the case of alkaline extraction, the high pH environment breaks down cell structure and solubilizes the proteins. And then with the isoelectric point precipitation part, we are precipitating the proteins and separating them from the material. In the case of microwave-assisted extraction, it uses dielectric heating, which causes rapid vibration of water molecules, breaking down the cell walls and releasing the proteins. In the case of ultrasound-assisted extraction, it uses acoustic cavitation, where microscopic bubbles expand and expand until they collapse, and they create intense shear force that disrupts the cell wall and enhances the protein recovery. And this is the general layout of my materials and methods. I did not include all the steps for all the experiments. However, this is the general layout, and it differs from treatment to treatment. And of course, there was optimization done for many different parameters like time, temperature, pH, and power. Moving on. I will be sharing some of my results. These are not all the results, but just the most important results. In the case of protein content and protein yield, since we are trying to see how these other methods, the non-conventional methods, can enhance the protein content and yield. What we noticed in my case is that in the case of tomato pomace is that there was no increase or significant increase in the protein content and yield. However, in the case of microwave-assisted extraction and ultrasound-assisted extraction, the major difference was the processing time. For conventional method, it took 1 hour, while for microwave and ultrasound, it took 3 minutes and 5 minutes respectively to be able to obtain the same protein content and protein yield. And of course, for a fair comparison, we also tried extracting at 3 minutes and 5 minutes for the normal conventional alkaline extraction, and we are not able to obtain the same protein content and yield as microwave and ultrasound. So why is this important, even though the protein content and yield did not increase? A shorter extraction time means lower energy consumption, higher possible scalability, and also reduce protein degradation risk. So a short processing time can help in general. And then moving on, another thing that we consider while extracting protein and for the final concentrate or isolate is the functional properties. Functional properties are very important for the applications that the protein extracts are going to be used in. And there are many different functional properties. Here I'm only showing 3. And for example, in the case of foaming capacity and stability, it is important in applications such as baked and aerated food products like whipped toppings. For water holding capacity, it is important for hydrated food products. And for oil holding capacity, it is an important indicator in foods where emulsions are necessary and fat-rich formulations will be produced. So these properties determine how the extracted proteins can be incorporated into different foods. And of course, as a comparison, they were compared with a very common protein isolate, which is whey protein isolate, in order to see how differently they react. They're acting. And then, as I continue my research, there are three key areas that I will focus on moving forward. First one being exploring more extraction techniques, then further research on both tomato pomace and tomato leaves, and finally evaluating food formulation applications. So the key takeaway is by refining extraction methods and studying their applications, this research can contribute to more sustainable and functional plant-based protein sources. So that's all from me. Thank you for your time. And if you have any questions, I will be happy to answer them in the Q&A session.
Thanks a million, Bodoor. It was a very, very nice presentation and very great overview. And I have a few questions as well later during the Q&A question. All right, this brings us then now to our third speaker, which is Özge Güven, also from the Middle East Technical University in Turkey. So, Özge, the stage is yours.
I'll share my screen. Could you see clearly?
It's there. The presentation is there.
Okay, then I am starting. Hello, everyone. I am Özge, and I will mostly talk about polysaccharide-based aerogels and their application in different food processes. But first, I want to introduce myself in detail. I complete my bachelor's degree in food engineering at Middle East Technical University. And at that time, I also completed a minor program in environmental microbiology. And last year, I completed my master's degree again in food engineering. Now I am PhD student in food engineering. And also I started to work as an researcher in my advisor's lab, Professor Öztop's lab, since my undergraduate years. And here is the project that I am included mostly. The first project is actually Aerogel project. We aim to design polysaccharide-based aerogels, and we wanted to apply them in different food processes. And the second project is actually the subject of my master thesis. We aim to design a refrigerator which is integrated with 405 nanometer LED light. And we aim to extend the storage time and food safety levels of foods. And I mostly work with strawberries in my thesis. And the last project is such a quality, and its aim is to develop alternative quality control methods for the sugar, chocolate, and confectionery industries. Within the scope of this project, I visited two countries, Chile and Germany. Within this visitings, actually, I trained a lot. I learned new techniques. I performed too many experiments also. And here there are too many photos from my research journey. I gained a lot of professional experience during all the tasks I completed during my journey, but also I gained a lot of life experience. I met a lot of people. I had never been abroad before, and my research journey gave me a chance to go to abroad. I had a chance to get to know different cultures. Some of my colleagues that I spend every day, all the time, become as my family. And also, I had the chance to work with a lot of different people with different perspectives. And actually, it is the best thing because it showed me that how science is such a collective. And how I met with ProxIMed's professionally is a perfect example of how science is collective, actually, because I'm mostly working with supercritical carbon dioxide extraction machine to produce aerogels. And one day, my advisor emailed me like, Özge, do you want to try something new? And then we met with Lisa and then Simon, and I performed for the first time the fetting process for them, for their samples. At the following month, my colleague Elif went to Lisa and Simon's lab as a visiting researcher, and Simon and Elif worked together. And this story always makes me motivated because I believe that not sharing or not sharing your experience not contribute anything, but sharing it contributes a lot, even if it's small. So now it's time for the aerogels, which is my major research area, actually. I want to first mention them briefly to clear the basic concepts. Aerogels includes almost 90% air, and they have high surface area, low density, absorption ability in a perfect way, and they are highly porous, they are ultralight, and they are also high in mechanical strength. And within its unique properties, aerogels use many fields, and this is the basic steps of to produce aerogels. If you want to produce aerogels, first you need to produce hydrogels, and you need to perform a solvent exchange by alcohol. And then you need to remove all the alcohol with supercritical carbon dioxide drying. And why supercritical carbon dioxide drying is that much crucial? Because the most important thing about aerogels are their porous structure and high surface area, actually. And supercritical carbon dioxide drying causes almost zero surface tension. And by this way, high mass transfer coefficient occurs, and the solvent included in the gel structure removed quickly. By this way, gel structure is perfectly preserved. And also, this technology is non-toxic, consumes less energy, and so we can call it as a green technology. And within Aerogel's unique properties, we want to get it advanced within its absorption ability. So first, we want to design our own aerogel production method, and we want to continue with the applications. Why we also choose polysaccharide types aerogels? Because they are natural, biodegradable, biocompatible. It's really safe to use in food applications. And as the first step, we optimize our aerogel production method, and successfully we produce our xanthan chitosan, and also sodium alginate aerogels. And within the first work package, we work with chitosan so much because its deacetylation degree is too important. So we also realize that this deacetylation degree affects our gel structure so much. So within this concept, we publish our first paper. And then we started our applications. The first one is regeneration of frying oil. Why we do this? Frying oil must be odorless, uncontaminated, and oxidatively stable. And the frying process stimulates the production of free fatty acids in oil, and they change the taste and quality and also lead to the formation of harmful compounds. And we believe that we can absorb free fatty acids within the ketone aerogels, and we achieve this. And this application is also the master thesis of Feyzanur Delice, and we also published one paper from this application. And our another application is clarification of cold tea. Why we do that? Because teas have unstable colloidal structure due to the protein, polyphenols, free amino acids, caffeine, and goes on. And when it's stored below 4 Celsius degree, these molecules become insoluble, and they are creating tea cream. And this tea cream causes turbidity and deterioration in color, taste, and appearance. So they should be removed from the concentrates. And in industry, in food industry, it's normally performed with magnesium silicate, but it's really hard to filter it from the concentrate. So we just wanted to provide another alternative. And we achieved to decrease turbidity of cold teas. And within this application, I had a chance to present our results in a FOST conference last year. And as a last application, we used Aerogels as a support in enzyme immobilization. We actually firstly work with pectinase enzyme because this enzyme is really valuable in food industry, especially in fruit juice industry. It is used as an again to clarify fruit juices, but these enzymes for just for the single use. So we aim to support its activity at the same time. We aim to use them a couple of times. And within this application, I had a chance to present our results in International Food Innovation and Sustainability Congress last year also. And we awarded first within this application. And here is our aerogel team. There are professors, PhD students, master students, and our lovely undergraduate students. I'm really thankful all of the people who contribute our result projects. We worked a lot, and thank you for listening to me. If you have any questions, I will be happy to answer.
Thanks a million, Özge. Very, very nice presentation, and it touched
Thank
my
you.
heart a little bit. So,
I know.
so thank you very much. And it
Mm,
is really
Thank
nice
you also.
to see how people are coming together and yeah, getting research further ahead. So thank you very much. And I encourage everyone again to note either a few questions, ask them during Q&A session or put them in the chat, and we will come back to them later on. And before we have a short break, we have our next speaker, which is Zülal Aksoy from Ege University from Turkey. And I open the floor for Zülal.
Okay, I am sharing now. Can you see my?
We can see it, but not in presentation mode yet.
Okay. It's okay now?
Oh, now it's fine. Yeah, thank you.
Okay. Hello everyone. Welcome to this webinar today. And I will be talking about, firstly, my name, Zülal Aksoy. And today I will be talking about exploring mallow leaves as a novel protein source using ultrasound-assisted extraction techniques. Firstly, who are we? We are a research team at Ege University working on the production of protein powder from mallow leaves, led by Professor Seda Arsus. This project is carried out by PhD students Zülal Aksoy at the Faculty of Engineering, Department of Food Engineering. About Ege University. Ege University is a leading research institution in Izmir, Turkey. It has a strong international reputation with over 1,000 national and 74 European-funded projects. And the university collaborates with regional and national stakeholders conducting over. And beyond education and research, Ege University plays a leading role in transferring scientific outcomes to society. And as a fourth-generation university, it strives to create synergy in the research and innovation ecosystem, contributing sustainability to science and society. About me, my PhD story. I chose to study food engineering because I have been passionate about discovering new things and conducting experiment science childhood. Later, I met my advisor, Professor Seda Arsus, and had the opportunity to be involved in many her projects. Throughout this process, we worked on exciting studies such as the production of vegan mayonnaise and protein powder from various leaves. Realizing how much I enjoyed this field, I decided to pursue a PhD. Currently, our research focuses on the techno-functional properties of a proteins derived from different leaf source, protein conjugates, and the development of a new vegan product. Why we select a mallow leaf? Mallow is a wild medicinal plant and grows spontaneously and requires no special care. It is rich in the protein, making a valuable alternative protein source and the most high protein content. The mallow leaf was collected from the garden in Izmir in November. About total dry matter content of mallow leaf was 21%, and the protein content of mallow leaf in dry bases found of 44%. The protein content of fresh leaves by determined by Kaldal method. Production of proteins from mallow leaves. You can see this is a main protein production flow chart, isoelectric precipitation, temperature, time, and pH value depending on the leaf type. And you will see our parameter study on this in the other slides. But this is the main flow chart. Firstly, the mallow leaf. After that, the alkali liquor and buffer solution. After homogenization and extraction and different temperature and time. After that, filtration, centrifugation, and we determine supernatant and isoelectric precipitation, pH value 3.5. After that, stirring and again centrifugation, and we use pellet. Lastly, freeze drying. Ultrasound-assisted extraction. Protein extraction from mallow leaves investigated of the influence of ultrasound on the protein yield. You can see how protein yield is calculated. And the firstly main flow chart, and we applied ultrasound before the extraction. About determining extraction parameters. These values were determined based on the results of our team's previous leaf protein studies and also utilized literature. About this table, conditions, parameters, results, we select the optimum parameters. For the temperature, solvent solid ratio, and ultrasound. You can see our parameters, and we select the optimum parameter, and we use for the extraction these parameters. And for this slide, comparison about two methods of which is the high protein yield. This select ultrasound-assisted extraction, the most protein yield highest is the 41%. You can see. To determine the effects of mechanical disruption on protein extraction from mallow leaves and increase protein yield, extraction was performed under optimum conditions with the addition of preheating and increasing the ultratrax by time by the 5 minutes. With this method, it was determined that the protein yield reached 60%. For about scientific contribution and impacts, this research contributes sustainability protein production, and the extraction protein can be used in food supplements and various protein-enriched products. Our goal is to introduce this alternative protein to Mediterranean consumers. Thank you for your participation. Thank you for your listening.
Thanks a million, Zülal. Very, very nice presentation. And also, according to green leaf proteins, I think we are on a great track with that, and I think they have a lot of potential in the future. So thank you very much for your contribution. Same as
And I want to something. Sorry about this, but I have left from the webinar now because I have another meeting started. And if you have questions about my topic, I wrote my email in chat. Is possible?
yes, perfectly fine. Thank you very much, Zülal,
Thank you.
for taking your time. And yes, if there are maybe you have five more minutes. If there are any questions right now, we could raise them. If not, I have a question. I saw you have pretty colorful protein extract. Do you have any plans on changing the color on those? Could you hear me, Zülal?
Sorry, I couldn't.
Okay. So you had colorful protein extracts. Is there any possibility, or how do you see you could decrease the color in your protein extracts? Any plans on that?
Yes, but we study now for this. But we want to obtain it protein more the whiteness, but on study.
Very, very good topic to study on. I can share some literature with you about the coloring, and yeah, because we have the same topics more or less with protein extraction from duckweed. So
Thank you.
thank you very much. Any other question for Zülal from the auditorium for now? Just double checking the chat. So we are actually quite ahead of time. I would like to do a break in a few minutes. Just asking, would it be possible, Mouna, would it be possible for you to present now and do the break afterwards, or would you prefer to have a break first?
I prefer presenting now, if it's okay.
Yeah, definitely. Okay, then I would like to introduce our next presenter. Her name is Mouna Hbaieb, and she's a a PhD student from the University of Sfax in Tunisia. Mouna, the stage is yours.
Okay, so I will share my screen. So you see the screen?
Yes, but not in presentation mode yet.
Okay.
If you start the presentation.
Now it's okay?
Yes, now it's right.
Okay. So first of all, I present myself. I'm Mouna Hbaieb. I'm first year PhD student in biological engineering at the National School of Engineers, SFAX. So my doctoral research focuses on harnessing the power of microalgae as a sustainable solution for alternative protein production in the future food industry. I have a master's degree in microbial biotechnology. So I'm truly honored and thrilled to have the opportunity to join today's webinar, sharing ideas, learning from brilliant minds. Really, events like this remind us how collaboration and innovation can shape the future of research. So, first of all, why the growing interest in alternative proteins? There are many reasons. First, the environmental sustainability. So alternative proteins such as microalgae often have a smaller ecological footprint requiring less land, water, and energy. Also, due to the population growth and the food security, health concerns, alternative proteins can be healthier because of the lower saturated fat acids and the fortified with nutrients such as essential amino acids. Of course, nowadays we have a lot of problems such as gas emissions, deforestation, and water overuse. So let's start with simple planning. First of all, we will start with the choice of microalgae, then optimization of biomass production, then determination of harvesting methods, biomass characterization, and finally the protein extraction. So this diagram outlines our work process. First, we begin from the culture of our microalgae called Formidium versicolor, a green culture and the lab scale. Then we move to the scale-up culture, semi-pilot culture of Formidium versicolor. Here we have the biomass separation, different methods of harvesting or separation. Then biomass characterization, its metabolites, protein extraction, and of course those proteins will be used in different will be incorporated in different application fields such as cosmetic, industrial, agri-food sector. So here 3 allophilic microalgae were cultivated at the laboratory scale in Erlenmeyer flasks. They belong to different classes of microalgae. For example, here Dunaliella salina belongs to the class of Chlorophyceae. Alomphora belongs to the Diatom class, and Formidium versicolor belongs to Cyanobacteria class. Here in this presentation, we will focus on Formidium versicolor, which is a cyanobacteria. We can call it also blue-green microalgae. So, with the aim of increasing biomass production, we conducted an optimization of the culture medium using the response surface methodology. Specifically, we selected carbon, nitrogen, phosphorus, and potassium concentrations as factors. So, the separation step is a sensitive stage, and there are numerous harvesting methods available. In our case, we selected the centrifugation and the sedimentation followed by centrifugation methods. So here, microscopic examination of the pellet and UV analysis of the supernatant serve as indicator of the cell conditions. That means the cell integrity. So here there is a comparative table of the two tested harvest methods. Various parameters were studied, such as, for example, recuperation efficacy, the loss rate, the cell integrity, and of course, the most important thing is the separation energy. So the sedimentation followed by centrifugation technique proved to be the most effective in terms of energy consumption and harvest time, while ensuring good cell integrity. With centrifugation speed of 4,000 RPM. So we then transitioned to the semi-pilot scale, referred to as scale-up, using an open photobioreactor called Raceway. As you see here in the photos, this system is equipped with a paddle wheel for steering, for agitation. Here, the biochemical characterization showed that Formidium versicolor, our microalgae, is rich with protein and carbohydrates. So this microalgae with a protein content of approximately 50% exhibits strong competitive potential compared to other protein-rich microalgae. Now we move to protein extraction stage. So two protein fractions were extracted, soluble proteins and phycobiliproteins. So phycobiliproteins are organized, as definition, are water-soluble pigment protein complexes which are found in cyanobacteria and certain algae. They are organized into phycobilisms and are classified into different types based on their spectral properties. So as I said, two different fractions of proteins, soluble proteins, and we extract soluble proteins using alkaline extraction followed by isoelectric precipitation. And for the second part, which is phycobiliproteins, we used the freezing and thawing method. We start with phycocyanine. Our phycobiliprotein is called phycocyanine. It's a blue pigment. So starting with the phycocyanine extraction, here our biomass of Formidium versicolor, we add the phosphate buffer. Then we did 4 cycles of freeze thawing to make sure of the cell disruption. Then this is the crude extract of phycocyanine. You see here the blue color. And finally, we do the phycocyanine filtration. We obtained a phycocyanine yield 11.37%. Here, ammonium sulfate was used as a precipitating agent. Different percentages were tested, ranging from 40 to 75%, and a UV visible scan was performed. So here, this diagram shows the different phycocyanine concentration for each percentage. The highest concentration was observed at 65%, and the highest purity index was obtained at 60%. Here, how do we obtain the value of the indexed purity? We do the ratio of the absorbance at 620 to the absorbance at 2080. And we will obtain the purity index, which is very important here, very important parameter for the phycocyanine. Now for the other proteins, other soluble proteins, after drying the biomass, ultrasonication was used as a pretreatment for cell lysis. Then for solubilization, the pH was varied using sodium hydroxide. We used the 3 pH value, 9, 10, and 11 for the solubilization. And after that, for precipitation, the pH was gradually lowered to 3.5 using a hydrochloric acid solution. So as you see here, the highest yield of protein was found with pH 11, using pH 11, and also the highest protein recovery. The value is 71.42%. While when we used pH 9, we obtained the lowest yield and the lowest protein recovery percent. The protein recovery. So thank you so much for your attention.
Thank you very much, Mouna, for your insightful presentation. Before we do our next presentation, I will give you the opportunity to take a little break, as we are already ahead of time. I suggest that now it is 14:32, and I would suggest that we meet again at 40:45. So 14:45 German time. This means 13 minutes break now. You can grab a coffee, grab a drink, yeah, do a biological break, and then we will be back at 14:45. So 12 minutes from now. Thank you very much, and see you in a few minutes. All right, so let's continue after our short break. Our next presenter is Dilara Ozcan from Middle East Technical University from Turkey. And Dilara, the stage is yours and you can share your slides.
Thank you. Sorry. Can you see?
Yes, now we can see your presentation.
And hi everyone, my name is Dilara Özcan and I am master's student in food engineering at Middle East Technical University. My research focuses on protein extraction and characterization from faba beans to explore their potential as a sustainable protein source. So before I dive into the details of my research, I would like to talk a little about the path that's brought me here. I started my food engineering studies at METU in 2016, where I built a solid foundation food science and processing techniques. In 2021, I had my first real research experience at TÜBİTAK as an undergraduate researcher, working on a project related to the extraction of emulsifiers from chickpea aquafaba. It was my first deep dive into the physical-chemical analysis and food formulation, and it really sparked my interest in food science. In 2022, I continued my research journey at METU, this time focusing on bioactive compounds from olive pomace, a project that got me thinking more about sustainability and using food byproducts in a meaningful way. Then in 2023, I became a part of ProxIMed's project where I worked on plant-based protein extraction from faba beans. This project has been an exciting challenge, giving me the chance to experiment with different extraction techniques and optimize protein functionality for plant-based foods. And beyond the technical skills, ProxIMed has been an incredible learning experience, not just in research, but also collaborating with experts and sharing knowledge and seeing how food science can contribute to a more sustainable future. Now let's talk about the faba beans, which is the core of my research. Faba beans are ancient and nutrition-dense legume, also known as horse beans or broad beans. They rank after They rank third after soybean and pea in importance among food legumes and widely used in different forms, for example, dried, soaked, roasted, or canned. Nutritionally, they are rich in proteins, approximately 20 to 41%, with a good balance of albumins and globulins. And their low lipid content and high lysine content make them an excellent plant-based protein source, especially for protein extraction. Most importantly, faba beans are cost-effective and sustainable alternatives for plant-based diets. Let's talk about why we are focusing on faba beans. The goal of this research is to develop high protein faba bean concentrates for plant-based diets. To achieve this, we worked with different varieties, dry, fresh, and residual canned faba beans. Compare their suitability for protein extraction. We explored two main extraction techniques. These are alkaline and enzymatic methods. And we assessed how different pretreatments impact protein properties. During this process, we introduced bleaching to improve the color quality of the faba bean protein concentrates. To enhance extraction efficiency, we tested different processing technologies, including microwave and pressure cooker treatments, to see their effects on protein yield and functionality. The final step was to optimize extraction conditions using central composite design or response surface methodology to maximize protein yield and solubility. And by combining these approaches, this research aims to make faba bean proteins more functional and more suitable for plant-based food applications. Let's talk about our progress. This diagram represents the current progress of my research. We have so far focused on dried faba beans, testing alkaline extraction with bleaching, pressure cooking, and microwave treatments, while enzyme-assisted alkaline extraction still in progress. Here is the workflow of my study. We are starting with different types of faba beans, applying pretreatments, and followed by alkaline and enzyme-assisted extraction, and then freezing, and finally analyzing extracted proteins. Here you can see alkaline extraction process. The alkaline extraction method, a widely applied technique for protein recovery from plant sources. And dried or bleached faba beans were selected as the raw material, and different pretreatments, including pressure cooking and microwave treatments, tested prior to extraction. The case step is to adjust pH to the alkaline level for protein solubilization, and then the pH was lowered to isoelectric point to induce protein precipitation efficiently. The pH shifting method is really crucial for this alkaline extraction methods. And for this study, initially two pretreatment methods and two extraction techniques were planned to for application to three different faba bean varieties. However, first trials conducted on untreated dried faba beans with alkaline extraction, there was a significant issue. The extracted protein has really undesirable dark color. To address this bleaching methods introduced as an additional pretreatment methods. As shown in the images, you can see bleaching significantly lightened the protein, making it more suitable for food applications. However, when we look at the protein content results, bleaching increased the total protein content, but for the soluble protein content, bleaching really decreased to soluble protein content. When we combined pressure cooking and bleaching, the decreasing really significant. This suggests that structural modifications occurring during these treatments may have reduced the protein solubility. When we look at the amino acid profiles bleaching didn't significantly reduce amino acid levels compared to the control, and also overall protein quality is preserved. Also, increase in certain essential amino acids is observed, suggesting that bleaching may even enhance the availability of specific amino acids. When we look at the water holding capacity and oil holding capacity of the extracted proteins, the results indicate that bleaching alone slightly reduces water holding capacity compared to the control. However, when we combine bleaching and pressure cooking, significant increase in water holding capacity is observed, suggesting structural modifications that enhance water retention. In contrast, oil holding capacity remains relatively stable across all treatments. When we look at without bleaching, the aim was to evaluate the impact of thermal pretreatments on the color of the extracted proteins. The results indicate that pressure cooking increased lightness, making the product brighter compared to the untreated samples, while also slightly reducing redness and yellowness, resulting in less intense color profile. Although pressure cooking improves the color, the lightest and most visually appealing result is still with bleaching. While various pretreatments and extraction methods were applied to improve protein yield and functionality, several challenges were encountered during the process. One of the main issues was the milling effect. By reducing the particle size, it made protein extraction more difficult. Additionally, factors such as bleaching, pressure cooking, and temperature led to excessive protein denaturation and further hindering extraction efficiency. Another significant challenge, and this is really very big challenge, was starch gelatinization, which caused the proteins to become difficult to recover, ultimately reducing overall protein yield as seen as video. These challenges highlight the need for process optimization to balance protein recovery and functionality, ensuring the most efficient extraction approach. And this is the most funny part. Beyond its academic contribution, ProxIMed has been an unforgettable journey filled with a lot of experiences, challenges, and moments of learning. This project is not about research and innovation, but also about the friendships and memories. From long lab hours to inspiring discussions and laughter shared outside of the work, this experience have made this journey truly special. And with that, I would like to say ProxIMed for the friendship that lasts. Thank you all, and thank you for listening. If you have a question, I can take.
Thank you very much, Dilara, for this nice overview, and also for the heartwarming pictures at the very end. Much appreciated. I'd like to introduce our next speaker before, and therefore it's actually my colleague Lisa Ziegltrum from the University of Applied Sciences Weihenstephan Triesdorf, and the stage is yours, Lisa.
Thank you. So, I need the first page. So can you see my screen?
Perfectly fine, yes.
Perfect. So hello everyone, and a warm welcome also from my side. So my name is Lisa Ziegltrum, and I'm a PhD student at the Technical University of Munich and the University of Applied Sciences Weihenstephan Triesdorf. My topic is the protein extraction from lentils with different innovative and green technologies. And today I would like to talk about the ultrasound pretreatment effect on protein extraction from yellow lentils, bridging sustainability and efficiency in plant-based proteins. But before I tell you more about my current research, I would like to talk about myself and how I came to do my PhD in this field. So after I graduated from school, I heard about the food technology program and found the field very interesting. So I did my bachelor's degree in food technology at the University of Applied Sciences Weihenstephan-Triesdorf, the HSWT. And the bachelor's program includes an internship semester. And therefore, I spent 6 months at the private dairy Bauer in Wasserburg. And there I worked in the quality assurance. And I really enjoyed it there. And I was very enthusiastic because I was allowed to see all areas. So I saw different productions. I was in the quality management, and I was even allowed to drive along to collect the milk from the farmers. So for this reason, I wanted to continue my education in this field and started my master's degree in food quality with focus on product safety, also at the HSWT. And there I took a subject, it was called research project, in which I extracted proteins from the stinchen nettle and tried to decolorize the powder as a protein drink was to be made from it. And here you can see an example of the powder that could be generated. And this was a lot of fun, and it sparked my interest in alternative proteins. So I wanted to do my master's thesis also in this field and with alternative proteins. And so I collaborated with the protein distillery, and they produce proteins from Bure's yeast with various functions. And yeah, I compared this protein with other alternative proteins and also in combination and determined the functional properties. And during this time, I was given the opportunity to do my PhD in the ProxIMed's project. And yeah, this topic was the perfect fit for that I was interested in. So after my master's degree, I started my PhD in cooperation with the Technical University of Munich and the HSWT. Yeah, so far we've been able to experience some very exciting things. So last year in March, we had a training school at the Ege University in Izmir, and there we baked protein-rich cookies and a lot of other things. In September, we had our face-to-face meeting in Porto, and here you can see that we enjoyed the stay there and the time together. And in November, we were at the EFOST conference in Bruges, and there I had my first poster presentation, which was also a very great experience. So you can see, and you saw at the other presenters showed you that you can experience very much in this time. You have a lot of fun, you meet new people, making friends, and yeah, this is very valuable. And now I want to show you where I'm working. I'm working at the HSWT in Weihenstephan-Freising, especially at the Institute for Food Technology. And yeah, not only the Institute for Food Technology, but also the Consumer Science and the FSIWS Food Startup Incubator is involved in the ProxIMed's project. And we at the Institute of Food Technology, we are working with lentil, chia seeds, and duckweeds, and using different pre, post, and protein extraction techniques, which we compare and analyze product nutrition, safety, and health aspects. And today I want to talk about the lentils and ultrasonication as pre-treatment step. Okay, so why lentils? Lentils have a great potential as an alternative protein source. They are recognized as a sustainable crop due to their ability to fix atmospheric nitrogen, thereby decreasing the need for fertilizers in cultivation and improving soil quality, and also require less water. They also offer nutritional benefits, including cholesterol and lipid lowering effects, as well as potential positive effects associated with a lower risk of chronic diseases such as cancer and type 2 diabetes. Their high content of phenols, flavonoids, and condensed tannins give them a superior antioxidant capacity. The lentils already contain 20 to 31% of proteins and have all essential amino acids. They have good functional properties such as solubility, foaming, emulsification, gelling, water and oil absorption capacity, and they have good sensority properties. In addition, they has an elevated digestibility, and overall, the diverse functions of lentil property allow it to be integrated into innovative food products. So lentils have considerable potential for use in dairy alternatives, and also in gluten-free baked goods and meat. And after the protein extraction, the effectiveness of the various techniques is elevated according to the protein yield, protein content, techno-functional properties, and the potential applications in the food industry. And our aim is to obtain a protein powder with a protein content more than 60% or specific techno-functional properties for a powder mixture for the production of meatball analogs. So at first, I want to talk about the conventional extraction, the alkaline extraction, and isoelectric precipitation. And for this, the pH value is first increased with alkali, and then the proteins are negatively charged and repel each other. And that makes them easily soluble. And if the pH then lowered to the isoelectric point, the net charge is 0, which means that the proteins no longer repel each other and aggregate and precipitate. And yeah, the alkaline extraction and isoelectric precipitation are examined separately from each other. So I started with the alkaline extraction, with the optimization of it first. And to do this, I used the response surface methodology, in which a test plan is created for me. And I specified the minimum and maximum values for the parameters to be analyzed. And then I got the trial plan. And I wanted to optimize the pH, the concentration, temperature, and extraction time. So for the alkaline extraction, the lentil flour was stirred at first in water in the tested concentration for 15 minutes. Then for 50 seconds homogenized with the ultra-Turax. And then the pH, temperature, and the extraction time were set. And to separate the soluble proteins from the insoluble components, there is a centrifugation step. And then I got my supernatant with the soluble proteins. And now when you see at the first table, you see the optimum values I got from the response surface methodology, the RSM. So the optimum values was calculated are pH 11, concentration 0.5%, temperature 65 degrees, and time 30 minutes. But I adjusted these values a bit because, for example, a pH of 11 leads to poorer sensory properties, and to generate more protein more quickly and use less water, I set the concentration to 5%. And to avoid denaturation, I also reduced the temperature a little bit to 50 degrees. And at the second table, you can see in the first line, the protein yield fit. This is calculated by RSM. And then you can see the real protein yield and the real protein content. So we achieved a protein yield of 92% and a real protein content of 38% with the optimum values. And with the adjusted values, we got a protein yield of 92% and a protein content of 60%. And on the diagram, you see that with the adjusted values, we could achieve a significantly higher protein content, and the protein yield don't differ. So you can say that the adjusted values are more suitable for the application. And one thing I want to mention also is that the standard deviation from the optimum value from the protein yield is very high, because the pellet I got after the centrifugation could not be separated properly. So it's a little bit difficult. So we see that the adjusted values are much better. So the second step was the isoelectric precipitation. And to optimize this, the adjusted values for alkaline extraction were used before. And after the first centrifugation, the pH, temperature, and time were then adjusted, and the sample was centrifuged again. And here the pellet is of interest with the precipitated proteins. And in this step, we got optimum values. You see at the first table, pH 5, temperature 4 degrees, and time 30 minutes. And here, to make the process easier and require less energy, the 4 degree was also adjusted to room temperature. And if you compare the protein yields again here, you see that there is no significant difference, but I could achieve a significantly higher protein content. So here we can also see that the adjusted values are more suitable and achieve better results. Now we move on to the ultrasound-assisted extraction, and I investigated this technique as a pretreatment step for conventional extraction. And what is ultrasound doing? So the ultrasound creates bubbles which become larger and larger until they reach unstable size and collapse. And this breaks the cells, and the particle size becomes smaller and the protein can escape. So this makes the protein extraction easier. So in general, ultrasound can reduce the use of chemicals, water consumption, and energy consumption, and also reduce waste. It increases efficiency and can save time. So it can lead to a more environmentally friendly production. And my aim was to be able to lower the pH value using the ultrasound, thus also reducing the use of chemicals and requiring a lower temperature. So for this, the calculated optimum values were pH 10, temperature 50 degrees, an amplitude of 100 and an energy input of 2,500 watt seconds per gram dry matter. And here the theoretical protein yield of 79 promised us a higher protein yield than with the conventional extraction, but this could not be achieved in practice. So we got a protein yield of 71%. And yeah, we can say that it's significantly higher than the protein yield from the conventional extraction, but it is only 2%. And the protein content of these both is nearly the same. So this means that I was not able to reduce the pH and the temperature of the time being. So further research must be carried out to see whether other energy inputs are still useful or whether the same results can be achieved with lower pH and temperature as with the conventional extraction. So now I would like to advertise briefly in case some students are there from the HSWT. So we offer final thesis on protein extraction from lentils, duckweed, and chia seeds and product development. So if you feel addressed, the topic is interests you and the lab work is fun, then please contact me. And now I want to say thank you for your attention, and you're welcome to ask me a question in the Q&A session afterwards. Thank you.
Thank you very much, Lisa, for your input. And now I have to ask if Andreas is there already, because the timing is a little bit off.
Oh,
So,
here.
okay, there he is.
Hi there.
Perfect. Okay, then our last presenter is Andreas Guess, who is also a PhD student at the University of Applied Sciences Weihenstephan-Triesdorf, but also working for one of our partners, Green Survey, within the project. And yeah, I kindly want to give the stage to Andreas, and you can share your screen.
Yes, thank you, Simon, and thanks for all the presenters. Now let me quickly see if is this working. Can you see my screen?
Not in presentation mode yet.
Yes, but in general, yes.
In general, yes. Yeah,
Very
perfect.
good.
Works.
All right. So hello, everyone, also from my side. I'm going to talk about something a bit different, maybe a bit off for all of you. I'm going to talk about LCA life cycle assessment or sustainability assessment in general. As you already said, my name is Andreas Guess, and I work for Green Survey, also a partner within the ProxIMed's project. And yeah, first of all, I want to introduce myself. I've been working in LCA for 7 years at the Department of Life Cycle Engineering at the University of Stuttgart. And since the ProxIMed's project started, I'm also working for Green Survey. I did my my master thesis at the University of Stuttgart as well. I started out actually being an environmental engineer, so that focus was more on water supply system, on energy technology, all those classic engineering topics. But at some point, I sort of fell into sustainability assessment, and actually gained quite an experience there. And I think I'm also quite happy to have taken this way. My master thesis was on the effects of grazing sheep on climate, biodiversity, and land use. And after this master's thesis, I started working full-time as a researcher at the university. And since then, I've been working on projects on renewable materials, on energy provision, on automotive engineering, in food provision, so various topics. And of course, there is when you do a life cycle assessment, or sustainability assessment, you assess the processes that are existing, that are newly developed, but you also do method development. So you are trying to develop new methods of sustainability assessment. And this is also something that I'm doing both for Green Survey as for the University of Stuttgart. So I'm aiming for PhD within the ProxIMed project, with my focus on including protein quality into life cycle assessment. So this is my general topic, but I guess since most of the attendees here probably are not that familiar with sustainability assessment and LCA, I want to quickly give a introduction into sustainability assessment. So when we are talking about sustainability, we always think of the 3 pillars of sustainability, or a triple bottom line. So it is not only about being environmentally conscious or addressing environmental benefits or disadvantages, but also looking at social levels and economic levels. So a product or a process or a service should also be profitable from a monetary side, and it shouldn't be of any disadvantage on a social side. If we now look at how all these things come together, so if a product is only environmentally friendly, then it doesn't mean that it's necessarily completely sustainable, because if it doesn't make any profit, it won't be sold. If it is socially not or morally not acceptable, it should also not be produced or conducted, etc. So all these things always play together. When we are talking about life cycle assessment, we mostly talk about the environmental part, and we always look at processes and services from a really abstract angle. So we think of the value chain, or the chain of production that you see right here in gray, where you have all these different life cycle stages. Here it is shown in a clear line, but then again, if you have recycling and reuse, you can also talk about circular stages, but for this purpose, it is a line. But what you can see, we start out from the beginning, from the raw material extraction to manufacturing. We include transports, we include construction. We then can look at the use and maintenance of services, of products, then to the demolition or the disposal. And all these steps require materials, they require energy, they require water. And then, of course, then they also have outputs, so they generate waste, emissions, and byproducts. And all these inputs and outputs are gathered, and in each of these, the material generation, the generation of energy or water, they require or they create certain emissions, certain impacts on the environment, and this is what we calculate. And in the end, we get a product, and then we can calculate how much impact often it is calculated in equivalents, most famously probably the CO2 equivalents. So how much of these impacts actually is emitted for the production or for the conduction of one service. I already said most famously is CO2 and CO2 equivalents. So the effects on climate change. And these greenhouse gases that are emitted are very important and very important to calculate, but it's not the only thing that we look at. So you can also, especially when you have food technologies, it makes a lot of sense to look at land use, eutrophication, or acidification, but also resource depletion is a very important part, water depletion. We can look at various toxicity effects. And this, of course, then depends on the product that you're looking at, what is of interest for you, what is
Andreas, sorry to interrupt you.
maybe in this case not of interest.
Andreas?
And you have different positions. Yes?
Sorry to interrupt. Is it possible for you to switch off your camera because I guess the internet connection is not as good. Maybe it gets better when you switch off your camera.
Yes.
Let's check. Can you hear me well? Andreas?
I think he dropped off.
Let me check for participants because it's still okay. So now he's totally gone. Oh, okay. Are you back, Andreas?
Yeah, apparently my internet connection. I'm very sorry, at a hotel at the
No, it's
moment.
fine. If you just switch off the video, it should be
maybe
better.
yeah.
Because the sound was,
Let's
I would
try
say,
that.
95% of the time clear. Sorry to interrupt. So you still have time. Maybe just switch off the camera and you can share again.
We'll do that, and very sorry for the bad internet. Let me share my screen again.
Yeah, it's already better.
There you go. Now. Oh, so yeah, we are looking at various impact categories, and at the moment we are also looking at developing new indices like land use, like biodiversity, or the impacts on ecosystem services. So there is still a lot of place for development. And actually, I now I don't want to show you any results of mine because this probably will be a bit boring for you at the time. But I want to say, why did I choose this field, and why would it make sense maybe for researchers and technical assessment or sustainability assessment. So for me, environmental conservation is essential.
Andreas? Andreas, can you hear me?
Can hear me.
Now it seems you to be back. Do you have much more slide? Is he gone now? That's unfortunate.
All right, I can try one more time. I switched the Wi-Fi now.
Okay, perfect.
Let's see. I'm really sorry.
No, you sound better. You sound better.
Well, that's good. That's good.
Sorry for interrupting you that often, but there's no point. But you sound better.
Nothing to be sorry for. Let's get through it quickly. I'm almost done, so maybe the Wi-Fi holds for as long. All right. So why did I choose this field? Yeah. So I have the motivation to actually do something for environmental conservation. And within this field, I have the possibility to say if a process is ecologically sound, if it doesn't make sense, if there is something to optimize. So I think that's a really, really good point. And also from a career perspective, it makes a lot of sense because the importance of ecological considerations is huge, and in EU projects, you have generally low chances of getting a grant without the consideration of an ecological advantage. So it is always considered when you do new undertakings, and well, the EU wants LCA, life cycle assessment, as the method of choice for these undertakings. What's also interesting, I think, is that you get glimpses into all kinds of industry and academia. So I think what also my fellow presenters and project partners already mentioned is the exchange that you have with all kinds of researchers from various fields is just it's really, really good, and it's a lot of fun, and you can learn immense amounts of things. So, for example, in this project, I got many new. I gained a lot of knowledge in the field of food processing and food engineering. So that is just something that I really, really cherish. And now, as I just also heard from Lisa before, if you want to do a bachelor or master thesis or a student project, then this is very much possible. And we already had 2 master theses that turned out really, really well. And the general process of an LCA can be learned within the timing of a master thesis. That is no problem. And so if you're interested, here's my email address. And I guess we're going to share the slides later on, maybe? Yes, we are. So great.
Yes.
Perfect. And with that, I'm already done. Thank you so much for your attention. Maybe then another thing that's positive for working in LCA, like you get to do a lot of international traveling as well for these projects, and that also is a lot of fun. So unfortunately, sometimes the internet connection there is not the best, but still, thank you for your attention.
Thank you very much, Andreas. Well, we managed it anyways, and it wasn't too bad. I would say so. So thank you for taking the time and the effort. So as our presenters finish their presentations, I am happy to go to the question and answer round, and I saw quite a few questions in the chat. But I want to give the opportunity first to ask questions directly. Otherwise, I will start and transit to the chat. Is there any questions so far from the auditorium?
I keep my questions this time because it's a platform that you should discuss and you should just asking questions. So
yeah,
no questions from my side.
Perfect. So I actually have a few questions. I have few questions. So my first question would be to Özge. You mentioned a lot of traveling during your time and during your projects. Maybe it's a bit of a private question, but how do you make yourself a home when going abroad? How is it to travel as much?
Actually, for the first time, when I traveled for the first time, I thought that it will be really hard for me because it was also for the long time. I stayed 2 months in Chile, but it was really joyful for me because how can I say that? I connect with people deeply. And Chile also was really far away from Turkey. Like I almost went 40 hours flight. But uh, maybe because of this, I connect with people deeply, and I enjoyed. I didn't feel alone. I don't know. Maybe it will change in time in another travels, but for that time, I just enjoyed. I meet with people. I learn their culture. Sometimes I get shocked about something different, but no, I didn't feel like I am alone or it's hard. It was nice.
Okay, so you were able to make yourself a home even if you're not in
Yeah, I
Turkey.
guess so.
Great. Thank you, Özge.
Thank you for your question.
Very welcome. My second question would be for Bodoor. Is it how do you measure the effectiveness of microwave assisted extraction? Is it for yield, or do you use any transmission coefficients, or is there any method that you are using that you use to measure how good the extraction is with a microwave assisted extraction?
In my case, my main focus, I focused on comparing the protein content and protein yield, so purity and yield, and that's mostly it, protein content and yield. And also, I will be checking the functional properties for the extracts. However, in terms of comparison, since also I am mainly looking at the protein content and yield, I'm considering that. So I'm measuring the total protein content using CalDAO, and also I am measuring the soluble. I'm comparing those.
Okay. The internet was shortly gone, so
I realized that.
you mostly compare protein content and protein yield. Okay.
Mhm.
How is microwave assisted extraction yeah, benefiting? Would you say it's benefiting up to
I
now?
mean, I was hoping that it would give me a higher protein content or yield. However, it did not, but the processing time is much shorter. In the case of conventional, I tried different times, and to be able to reach to the same protein content that I got in the short time in microwave, it took almost an hour. It took an hour. But in the case of microwave, I was able to achieve the same protein content in protein yield. Actually, the protein content was even higher in 3 minutes. So my internet connection.
Well, I got most of it. Well, I guess we should connect you then with Andreas to see and to compare it with an LCA.
Yeah, maybe together we can get one internet connection together.
Okay. Another question would be for Sultan. So you were talking about bioreactors. Um, could you explain a little bit more in detail why it is important to use those reactors? Why you can't use just like any vessels, or why is hygiene as important?
Okay. Because we are using a sensitive microorganisms to grow in a really controlled environment. So before using bioreactor, I was trying my experiments in LMR flux. But when you conduct fermentation in LMR flux, you cannot control the pH and dissolved oxygen for your microorganism. So in time, during the fermentation, the carbon dioxide it started to generate because it's closed loop fermentation system. And if you cannot add air from the outside, That means that there will be ethanol production inside. So we don't want ethanol production in our case because we want to grow our biomass more and obtain protein from that. So we need air. That's why bioreactors can provide air and you can monitor your dissolved oxygen to grow your microorganism. And also, if the pH changes, you can add directly acid or base from outside because when you start the fermentation, you all sterile all of them before starting. You autoclave them all together and connecting them in a sterile condition. And during fermentation, you're not touching anything, and it directly goes and arranging itself.
Well, very yeah, specific topic, so to say, and you have to be very careful because otherwise, yeah, you
Yeah.
will not know what is growing in there after you open the reactor again. So therefore,
Yes, especially if you are working with bacteria, it's really hard to keep it in sterile condition. But yeast are better than bacteria. So if you visit our lab, I'm very glad to show you how I'm conducting the fermentation in here. So if you have a chance to come here, we are very glad to welcome you here.
Thank you for the invitation. Okay, this brings me to my next question. Dilara, you were talking about the yield from faba bean and that the yield was actually going down with bleaching and pressure cooking it. Um, Maybe the question is a little bit too early, but um, what's your idea on how to get the yield up in this case? Because you want to bleach it and also have a great yield. Any ideas on that so far?
And nowadays, I am working on optimization of bleaching process and really stirring time and also the hydrogen peroxide percentage is really effective. And also, again, I am using central composite design to learn which method is the best for protein yield. And nowadays, we conclude some results, and I guess if we can change the stirring time and also concentration, we can increase protein yield, but also milling effect and bleaching and pressure cooking. If we combine all of them together, uh, we can control every steps. And because of this, I am just checking how we can reduce these bad effects to change. For example, I am trying to increase particle size for milling, and also I am changing pressure cooking time, and I think we can achieve the best with this changing, but it's really on a progress now. But for the bleaching, we observed optimum results, and we did RSM with them, and I think, yeah, I think we couldn't reach to control one because the control one is the best, and but for the end product, protein content is lower than to bleaching one. Bleaching really increase the protein content. For this process, we should decide protein yield importance and also final protein content importance, and we should find the best way with that. And we are focusing on this nowadays.
Thank you
And
very
also
much.
maybe enzymatic progress can help us to achieve this.
Thank you, Dilara. Very good answer. Yeah, it's sometimes you have to take a trade-off. May it be color, may it be yield, may it be total protein content. So thank you for having a look into it. Another question for Andreas. So as you are a specialist in LCA, how important do you see alternative proteins to incorporate it in Mediterranean diets and in diets in general all over the world. Because you said, okay, it might get a little bit boring talking about numbers, but how do you see it in general, the whole topic about alternative and plant proteins?
I think it's absolutely vital to talk about this because right now our main protein source is meats and animal products, and especially meats from yeah, beef or ruminants are having huge effects on climate. Also, the effects on the environmental effects of animal husbandry on land use, eutrophication, water use are way higher than any alternatives. For example, a recent study on Fraunhofer study on wheat protein. No, it was soy. Suggested that the production of soy emits around 1.2 kilograms of CO2 per 100 gram of protein, and probably for a egg protein it would be about 5 to 10 times this, and for the protein of beef it would be even more. So a kilogram of beef has around 25 to 50 kilos of CO2 emissions, and that is not the protein counted. So those emissions are really high, and I think they might be just the future, because we are now, as I said, most of people are focusing on climate change, or focusing on greenhouse gas emissions. But especially in food production, land use is a big problem, because we not only want to use the area for food production, we also want to use it for energy provision. We need it for urban areas, and we also need it for biological reserves and nature reserves, which is an important point that just requires a lot of space. And this is sometimes that is often not considered.
I agree. Yeah, thank you. Thank you very much. So the last question I would raise, if there are no further from the audience, is actually from Faisal to Lisa, and we can discuss about it now. Do you want to open your mic, Faisal?
Yes, I also want to ask one question from Ayşe Sultan, if we have time.
Yeah, we have time, definitely.
So maybe after your question, I can continue my two
Yeah,
questions.
go ahead, Faisal. You can.
Thank you, Sam. So, Ayşe, thank you so much for your good presentation. So, I want to ask that you said that you change your pH and during the fermentation process and all of these things. I want to ask, when you change your pH and you make the acidic environment, or you make the basic environment, or you make the alkaline environment in order to get the more fermentation. So did it affect the growth of microorganisms? Because these are the main organisms that basically initiate the fermentation process. And my next question is, do you use some kind of like the enzyme kinetics or enzymes to boost the fermentation?
Okay, let me start with the first question, the pH one. I'm not changing pH. I'm specifically keeping it constant, because if you start at pH 5, you need to continue at pH 5, because microorganisms started to grow at that pH. During my fermentation, I am not changing it. But if I use a Lemaire flask, I cannot control the pH. That's why I need to use bioreactor, because I can control it. That means I can keep it constant. So during fermentation, we need to keep it constant. For example, I'm using Saccharomyces cerevisiae. I know that it can grow at pH 5 really good. So I'm using pH 5. But if you are using different microorganisms, you need to check it first, which pH parameter you should use for it to grow. So that's the first question answer. Is it okay for you?
It's perfectly fine.
Yeah. For the second question, I'm using enzyme only for the hydrolysis purposes because enzymatic hydrolysis can generate reducing sugar. That means fermentable sugar in our fermentation media. So we need that sugar to grow our microorganism. In my fermentation, during my fermentation, I'm not using it because it can disrupt my cell walls. So my microorganism can stop the growing. Maybe immobilization of the enzyme can be taught, but I don't think it's necessary right now because enzymes also expensive product.
So we can call it this as a downstream processing and the upstream processing.
Yeah. Also, maybe you can use another microorganism to produce your own enzyme to continue your microbial hydrolysis, not enzymatic hydrolysis. That's you can start that fermentation step with microbial hydrolysis to eliminate the enzymatic hydrolysis. You can consider this one.
Thank you so much, Ayşe.
You're welcome.
Thank you very much. Last question was actually also from you, Faisal, about the digestibility of the proteins extracted from Lisa from lentil. I guess we can say our partner from the University of Parma will conduct digestibility trials, so we actually don't know it yet. But Lisa, maybe you can come forward if you already read something about in literature, or is this anything you looked into already?
No, not yet, really. So I'm waiting for the results from our partner. And yeah, you ask for the comparison to animal proteins. So in general, you can say that plant proteins is the digestibility is lower than from animal proteins, but the lentils one compared to other plant proteins is very good, I would say. But let's see what the results tell us.
Perfect.
And thank you for your question.
Thank you, Lisa, for answering.
Thank you. Thank you. Okay, so I will close the question and answer round now. Otherwise, if there is any question left, you can contact our speakers via mail who weren't able to stay until the very end of the webinar. This brings me now to the conclusion and my final words. So first of all, thanks to all of the presenters presenting their research journey and the work that they conduct within the project ProxIMed, and also sharing their results that they are working on. I think it's very, very, very interesting to actually see what hand-on research looks like. And therefore, thank you very much for your contribution. And I'm very, very, very proud of every one of you. And yeah, it touches me actually quite a lot. And I'm very happy to have you all.
Yeah, and I just want to say, sorry, after just distributing some hearts, but it's really thank you very much for all, for your work, for your hard work, and also, as well for this opportunity to really exchange. I think it's a great platform as we created, and even with or without this project, I think this community will continue.
Thank you. Thank you for your words, Islam. Well, now it gets a little bit less romantic because my final duty is I would like you to evaluate this webinar and how you felt also educated and informed, and therefore I kindly ask you to answer the survey I'm sharing into the webinar. I just have to open it. 1 second. It should be up by now. I hope everyone can see it.
It is up, yes.
Perfect. So those are 6 questions. And yes, while you answer the questions, Nietzsche previously shared our social media channels and also our project website. So if you want to stay in touch and want to stay informed about how the project is doing, you can follow us on our socials and visit our webpage frequently. And if there is any new informations and publications, they can be found there. So if you are interested in our project, you can stay in touch. Right, I see 12 from 17 people already answered. That's great. So anyone needs more time to answer the survey? Just checking. All right, I will close the survey in a few seconds. So thank you very much for taking the time. And it looks oh, can you see the results? Thank you. So I'm very happy that you enjoyed your time here and that we could disseminate a little bit of our research activities and also sharing the journeys of each individual. Therefore, thank you very much once more. And with those last words, I would like to close this webinar. Thank you, everyone, for your participation and for your questions. And special thanks to all the presenters. You did a very great job. Thank you very much, everyone.
Thank you, Simon.
Take care and
Thanks for organizing, guys, again.
very welcome. And have a nice weekend, and we'll be in touch.
Well done.
Bye.
Thank you,
Goodbye,
everyone.
everyone.
Goodbye. Bye-bye.
Goodbye.